Hi, this is Rethik, your biology subject matter expert from MemoNeet, and once again, welcome to the biology series. So, in this series, we will be discussing about the very important concept that is what the enzymes which normally requires for the replication, transcription, translation, and all. So, we are also going to discuss briefly about what do you mean by transcription. So as you can see here that the living cells and almost all the living cells, if we take an example of E. coli, the process of replication requires a set of catalyst, which is nothing but an enzyme. So we know what is replication in sense of DNA. So if DNA, which is double helix in structure, right? So obviously it will be in this way. So this double-stranded DNA will get replicated. So during the replication, it requires a catalyst, a kind of enzymes, which will make this replication process very smoothening. So the major enzyme or the main enzyme is referred to as DNA-dependent DNA polymerase. Okay, what is the enzyme name? It is DNA-dependent DNA polymerase. Since it has been used as a DNA template to catalyze the polymerization of deoxynucleotides. So as you can see in this figure, that these enzymes are highly effective. Okay, so that is the DNA polymerase that you are seeing is actually an highly effective enzyme as they have the catalyst polymerization of large number of nucleotides in a very short period of time. Say like if this is a single stranded DNA, now what you have to do? So you have to synthesize an other strand, which is complementary strand to these nucleotides. So if these are the nucleotides, you have to synthesize a complementary strand to it. So this DNA polymerase is a catalyst. It has large number of nucleotide which can synthesize polymerase. Okay, the large number of nucleotide in a very short period of times. That is what it is showing. That is addition or adding of the nucleotide. What kind of nucleotide? Not the random nucleotide, but which is complementary to the base pairs. So coming to the E. coli, we know that E. coli, it has only 4.6 into 106 base pairs. So when we compare that with humans base pair is more. That is, since it is diploid in nature, it contains 6.6 into 109 base pairs. Okay, so that means it's larger human DNA, like you need to synthesize there, it has larger when compared to E. coli. So what happens when competing with the replication of E. coli? It completes the process of replication within 18 minutes with this base pair. Okay. So that means the average rate of the polymerization has to be approximately 2000 base pair per seconds. Not only due to these polymerase have to be fast, but they also have to catalyze the reaction. It means that so during the polymerization, okay, adding of this nucleotide, the process has to be faster. This is first point. Not only being faster is important, but also it should, add the nucleotide in an accurate way. That means it should add a complementary nucleotide itself, complementary base pairs itself. And if there is any mismatch, wrong match happens, it could lead to, or what happens as a result of replication, it will lead into mutation disorders. That is what the point they're saying. That is, any mistake during replication would result into mutation. Clear? So for longer DNA molecules, if the DNA molecule is longer, since in DNA, two strands is present, right? So these two strands of DNA, cannot be separated in its entire length. Say like if you have this DNA now, okay, DNA is double-stranded, obviously now this is so big. You cannot separate the entire DNA in its one length. Okay, so to separate that DNA, a high energy is required. Okay, so which is not possible. So the replication occurs within a small opening of the DNA helix. referred to as replication for. Getting? So where replication will happen? Replication is going to happen within the DNA, okay, where within a small opening of the DNA helix, which is referred to as replication. Say, like if it is a DNA, what happens now? It starts moving opposite to each other. That time, what happens? And another complementary strand starts getting synthesized. Right. So we know a very important thing is, so yeah, so what happens is the DNA dependent DNA polymerase. So here the DNA polymerase, which acts as a catalyst, right? What it does, it helps in polymerization in one direction, only in one direction. Say like we have here the DNA. So if this is a DNA, there will be a replication fork here. So say like if it is 5 dash to 3 dash direction, and this is 3 dash to 5 dash direction, obviously. So here the replication will actually happen from this direction, one direction. Okay. Say like that is 5 dash to 3 dash direction. This creates some additional complication at the replication fork. So either you will be seeing two types here. One is continuous synthesis you're going to observe, and another one you are going to observe is discontinuous synthesis. So as you can see, this is a template DNA. What do you mean by template DNA? It is nothing but parental DNA. So till now you have parental DNA. Okay. So now this parental DNA will start getting moving. Say like if it is the DNA, right? So it will start moving opposite. So obviously in this you will see the replication fork. So from this region, a newly synthesized strand will start. That means newly RNA, like the another new strand will start to synthesize. Okay. So on one strand, that is the template strand, that is if you go with this 3 dash to 5 dash direction, what happens? The replication is continuous. So you can see here the replication is actually continuous from 3 dash to 5 dash direction. So obviously from this direction, that is from this direction, the replication is continuous. But there is another template strand is also getting synthesized here to the template strand, that is from 5 dash to 3 dash direction. So you can see that as a discontinuous strand is getting synthesized. So the discontinuity, after a synthesized fragment, later they get joined by an enzyme called DNA ligase. So totally how many enzyme that we have covered, we have came across till the slide. One is DNA polymerase as an enzyme. Then we have DNA ligase. What is the major function of DNA polymerization or DNA polymerization? Polymerase. The major function of the DNA polymerase is polymerization, addition of the nucleotide. Okay. And what is the major function of DNA ligase? It acts as a glue. Okay. To attach the discontinuous synthesis of the fragments. Okay. Moving to the next part. So that is the DNA polymerization on their own cannot initiate the entire process of replication. That is what they're saying. That means alone DNA polymerase will not initiate the entire process of replication. Also, the replication does not initiate randomly at any place. Say like if it is a DNA, obviously replication will not happen randomly in any place. There has to be a specific space. Say like in E. coli, if we observe, okay, so there will be a specific place. Same thing in the eukaryotes also. There will be a specific place from where this DNA replication will takes place. So there is a definite region in E. coli DNA where the replication originates or replication takes place. Such a region is termed as origin of replication. So such region is actually termed as what? Origin of replication. So it is because of the origin of replication, as you can see here, you have the origin of replication is a region. So from where the DNA replication will take place, the DNA fork will start occurring. So this is because of the requirement of the origin of replication that the piece of DNA, if needed to be propagated during the recombinant DNA procedure, requires a vector. So the vector provides the origin of replication. So what happens in E. coli? You will find a specific place. Like if you are choosing a vector or if you are choosing the E. coli DNA, so there will be a specific place called origin of replication. So from where this replication will takes place. The initiation process will occur in the origin of replication itself. Okay. Going to the next slide. Further, not every detail of the replication is understood well, but in eukaryotes, the replication of DNA takes place in S phase of the cell cycle. So in cell cycle, we have M phase, synthesis phase, then G1, G2, G0, right? So in the S phase, that is in the synthesis phase, we can actually study this replication. We can actually see this DNA replication takes place in the synthesis phase. So the word itself decides that synthesizing is happening. The replication of DNA and cell division cycle should be aligned coordinately. That is, during the cell division and the replication. Okay, that is, if the cell is getting divided, that is one point, and even replication will be happening, DNA replication, right? Both should coordinate with each other. Okay, should have been highly coordination. That is what they're saying. And during this coordination is failed, if a failure in the cell division after DNA replication results in what? A chromosomal anomaly is nothing but polyploidy, a kind of chromosomal disorder. The next very important topic is transcription. What do you mean by transcription? Right. So in the transcription, it is a very first steps of gene expression that involves in the formation of RNA molecules from the DNA, or another way of defining transcription is the process of copying genetic information from one strand of the DNA into RNA is termed as transcription. Okay. Say like if this is a one strand of DNA, so you are copying this information into what? RNA. RNA is nothing but single strand. So DNA will always be a double strand. RNA will be a single strand. From RNA, you will be finding mRNA. There are proteins, a messenger RNA. So this is termed as what? Transcription. However, unlike in the process of replication, so which one set is the total DNA of an organism gets duplicated. So if this is one DNA, it gets duplicated into 4, obviously. So in transcription, only a segment of DNA and only one of the strand is copied into RNA. So in replication, we know that if there is a one set of DNA, it gets divided into 2 now. Okay, the one set of DNA will get divided into 2 sets. But in the transcription, what we have to concentrate is their individual. Okay, that means the segment of the DNA. Say like this, if this is it's a single segment of the DNA, so that will be copied into RNA. That is what they're saying, right? So why does the strands are not copied during transcription? So here we have actually a simple answer for that. First, if both the strands acts as a template, say like we have DNA, right? So double-stranded DNA, where both the strand here, it acts as a template, they would code for RNA molecules with a different sequence. Okay, so you will also be going up with a sequence, like for this strand, you have ATGC, something like that. And for this strand, you will be having TACG, something like that. Okay, so you will see for this, as something different has to code, and for this again something different sequence has to code. And in turn, if they code for protein, the sequence of amino acid in the protein should also be different. Okay, hence one segment of the DNA would be coding for 2 different proteins, and this would complete the genetic information transfer mechanically. Secondly, the 2 RNA molecules, if produced simultaneously, would be complementary to each other, hence would form a double-stranded RNA. You're getting? So this is another point. Then this would prevent the RNA from being translated into protein, and the excess of transcription would become no meaningless in sense. So my major thing or a motto is, from the DNA, we are synthesizing the RNA to the RNA molecules and what the RNA is going to code for the protein. So if unknowingly, if in the DNA, if it would have code 2 different proteins, okay, and in one way, if the proteins are complement to each other, they're going to form an RNA strands, right? Double strands RNA. If they are similar and if they code, they're going to form what? Double-stranded RNA. So if they form double-stranded RNA, what happens? The protein synthesis will be prevented. RNA, it would prevent RNA from being translated into proteins. So if there is no translation will happen. So if there is no, what happens? If there is no transcription is going to take place, as a result, what we are going to see now? there will be no protein formation. That is, there will be no messengers now. So what is this transcription unit? A transcription unit in DNA is defined primarily by three regions of the DNA. So one is the promoter region, which is going to initiate the DNA synthesis strand. Then the structural gene and the terminator. Right. So first, since the two strands have opposite, what do you say that? Opposite polarity, and the DNA dependent RNA polymerase also catalyze the polymerization in only one direction. In sense, so can you see here that the two strands that you are seeing, one is in blue color and one is in the red green color, right? So the DNA dependent RNA polymerase, so they are having opposite polarity, like 3 dash to 5 dash, you can see here. So to this 5 dash you are seeing 3 dash, and here you are seeing what? 5 dash. So the polarity is opposite in direction. Okay. So catalyzation will also happens opposite to that. Getting? So say like if this is a parental strand, okay, if this entire thing is a parental strand and this is newly synthesized strand, will always go in an opposite direction to the polarity, opposite polarity. Okay. That is, so we have 5 dash to 3 dash, it is going. This is a strand which is that has the polarity 3 dash to 5 dash. So here 3 dash to 5 dash is acting as what? A template strand, and this is 5 dash to 3 dash is acting as a coding strand. Okay. So the other strand which has the polarity, that is what? It acts as what? A coding strand, a sequence sum as RNA. Okay. So the other strand which has the polarity, this is 5 dash to 3 dash, and the sequence is same as RNA, except thymine at the place of uracil. I'll say you what it means. So is displaced during transcription. Okay. Right. So, strangely, this strand, which do not codes for anything, is referred to as coding strand. How? Now, in this, diagram or the figure, you can actually see what you have been seeing. The strand here. So obviously 3 dash to 5 dash, right? And here 5 dash to 3 dash, which is what? A coding strand. So here it is a code. So this is a template strand and it is the coding strand. So A, T. So obviously what you are seeing, there is no, similarities. So that means this coding strand, it is actually becoming a kind of what? The point hypothetical sequences from the transcriptional unit. Okay, that is what it is referred below. That is the structure of transcriptional unit. So what happens here is we have the promoter here, right? Then what do you say that the terminator is also there, and the terminator flank the structural gene. So this flanks the structural gene in the transcription unit. So what happens here? The promoter is said to be located towards 5 dash end. So this is 5 dash end. This promoter is located at the 5 dash end, right? So from 3 dash, it is going to be in an opposite way. So it is located in the 5 dash end upstream region of the structural gene refers is this thing. Okay, so from where the coding strands will start. So from this region, the coding strands will start. So it is the DNA sequence that provides the binding transcription unit, okay, which is going to provide a site for the RNA polymerization. So if this is DNA, so this is a site from the promoter itself, you are going to see the RNA polymerization taking. So in the presence of this promoter, okay, which binds the site of the RNA polymerase, and in the presence of promoter, what happens? The transcription unit that we are seeing, right? The transcription unit, okay, it also defines as a template and coding strands. By switching its position, with the terminator. So terminator is the end position now. Okay. So where the coding is completed now, and the template that you are seeing strand could be reversed. So in transcription, we can reverse the coding actually. So the transcription is located. So this where you will be seeing now terminator. So terminator will always be located in the towards the 3 dash end of the coding strand. So it is being located at the end of the coding strand. That is 3 dash end towards the downstream. So where if you want to make it reverse transcription could also happen through the help of terminator. Okay. Okay. And terminator is actually considered to be end of the process. End of the process. That is synthesis process. It is at the end of the synthesis process. So additional regulatory sequence that may, so here we will be having additional regulatory sequence will also be there. So it may, present in future upstream or downstream to the promoter. So what happens? You have a strength of the DNA and you want to what synthesize an RNA, which is there obviously. So once the synthesis is over, so it will reach us to the terminator will be there. So it is going to stop the synthesis. Okay. If you want to make it additionally, you want to synthesize again the more, you will be having what? So additional regulators will also be present. That is what about the transcriptional unit, how exactly the transcription takes place. Okay. So how coding strands will takes place, how this template strand and coding strands will takes place, how from the DNA, okay, how the copying of the genetic material or genetic information from one strand of the DNA into the RNA is turned or RNA is happened, and from the RNA how the proteins or mRNA, okay, they are being synthesized. That is what we are going to study in the transcription. Clear? This is all about the transcription. And yes, you can contact me if you have any doubts regarding this specific topic through the comment section or through the Telegram channel. This is one point. And if you're planning to buy the Elite plan or Brahmastra test series or PYQ and coming, that is tomorrow, 25th and 26th of September, so the price of this entire Brahmastra test series and Allied Plan PYQ is going to minimize, that is going to decrease. So if you are planning to purchase any plans of the MemoNeet, you can check out tomorrow and after tomorrow, because the price is also getting lesser. So this is a very good opportunity if you want to buy any plan. And thank you and stay tuned with the MemoNeet.